Now we're going to continue. This time we're going to hear from 2cureX.
Thank you, Sven. One should think that 2cureX, that's because we're only two. We are more than that. And I would just start by saying that there's been two major changes since we met last year, where I gave the talk and where I was the CEO of 2cureX. And the one is that we have finalized a 3-year clinical trial, very important trial for us within metastatic colorectal cancer. And the other good news is that we have been able to attract Fernando Andreu as a new CEO of 2cureX that are going to push us to the next level. So, Fernando?
Thank you. Yeah, so pleasure to meet you. And it's a real pleasure to speak in front of real people instead of a camera. You know, that hasn't happened for the last 18 months, so I'm really excited to be in front of you. I just want to start by introducing myself briefly. I am Fernando Andreu, born in Barcelona, and I come from 35 years working in the in vitro diagnostics and medical devices industry. I have worked for large organizations like state 10 years at Roche Diagnostics, 10 years at Sysmex, which is a Japanese company. I have also worked for smaller organizations. I think what is relevant for us is in the last 15 years, I have been very much focused in precision oncology, and I will come back to that in a moment. At the same time when I joined, and that was 1st of February, we also appointed, I was going to say I brought with me almost, because I know Kenneth from the past, a new CFO as well. So, and this is Kenneth Johansen, and he has a very strong background in financials, of course, but also, and this is very important, he has a very strong focus also in business development. So he has been CEO of a couple of companies, startup companies, and also very relevant for us in the future, one of the last companies where he has been acting as a CEO is in the field of artificial intelligence and machine learning related to healthcare data. That's going to be very relevant for us. So, Ole said 2cureX was able to attract me, and I get this question very often, why did I join 2cureX? And it has to do with these 15 years that I mentioned about being in the precision oncology field. So precision oncology, I'm sure you're all familiar with it, but it boils down to two things. One is predicting how the patient will progress, and predicting which drugs will work on which patient. This is the summary of what we are trying to do with precision oncology. And the approach to precision oncology in the last 20 years has been very much focused in genomics and in general biological biomarkers. So everybody thought with the Human Genome Project we would know everything we need to know from a patient, which drugs will work, which won't, etc., etc. Reality is unfortunately much less shiny than this. It is a very limited number of patients who benefit from precision oncology based on genomics biomarkers. And I know that because I have been there. I have been selling genomic biomarkers, trying to sell other things, etc. And the truth is, trying to predict how the patient will respond based on the biology of the tumor is still out of the reach of the science, because we don't have a good model. The biology of the tumor is so complex that we don't have a good model of how a specific patient will respond. So for me, the answer to that is functional testing, where you take the opposite approach, which is, okay, forget for the time being the biology. Let's look in practice which drugs work or don't work in this specific patient. And then let's use that as a decision-making point. And that's what 2cureX is about, functional testing. So I had been looking at functional testing for a while, and 2cureX is one of the companies who is leading this space. So when I got the opportunity to meet Ole and other members of the team, for me it was a no-brainer to join. So that's and sorry for the personal story, but I think it ties well with the reason why we are here and the reason why we are here as a company. And I think the next slide illustrates a little bit more. Ole, you want to take this one?
Yeah, as Fernando mentioned, the traditional approach in designing the right treatment for the individual patient is to use genomics, molecular biomarkers, and also, as Fernando mentioned, well, I mean, we do have some problems. First of all, it has been done since we elucidated the human genome, so this is not a new story. But still today, more than 70% of patients, and this is a study in the US that's just come out, they do not have a genetic biomarker to guide their treatment. And here we are talking about single drugs. And you need to remember that in cancer, we almost always treat with combinations, and there the number of biomarkers are even lower. And this is a study that just came out. It's published by ESMO, the European Society of Molecular Biology, here in April. And what they saw was these 73% are illegible. So there is a biomarker for a specific drug. 27% are eligible, and the remaining part, almost 75%, there are no biomarkers. So if you take that piece of the cake and then look at that, well, being eligible is not the same as it works. So of those, only 11% respond to a treatment that is based on that molecular biomarker. And so what we say is functional testing is obvious here and it's obvious there. So does it work then? Well, let me just show you a slide where I introduce IndiTreat. IndiTreat is our way of personalizing cancer treatment. It's a cell functional test that can monitor both drug efficacy and resistance. And this is just the flow from the patient. This is a CT scan of a liver with metastasis. So a primary target for us is colorectal cancer. When the disease progressed to a metastatic stage, they will get metastasis in the liver. That's what you see here. We take a needle biopsy. From that biopsy, we can make hundreds, sometimes thousands of small copies. These are 3D copies of the patient's tumor. And we have a number of studies and publications showing that they resemble the tumor in vivo. They have the same heterogeneity, they respond differently, and the response is related to patient outcome. So then these microtumors, we call them tumoroids, they are then put into a screening system. And this is in essence very simple. What we do is that we follow these small guys. In this case for 11 days now, we'll do it a little bit shorter. We'll probably do it about a week. We need to follow our period, so we can simply follow how they grow. And if we find the right treatment, we will simply stop that growth. And that's, in essence, the test. It's a little bit what you have done for antibiotics since the 1960s, that we just want to find the drugs that kills that tumor. And we can expose it to single drugs, that I meant before, but even more important when we talk cancer treatment, it's combinations of drugs. And we see more and more combinations. And I would say I hardly know any cancer disease that is treated with single drugs. Some of them start with a single drug. I mean, immuno-oncology is a good example. There we hoped Keytruda could help patients with that one drug. We now treat combinations. PARP inhibitors were also these wonder drugs 5 years ago. Now they are also being given in combination. So that is what we are going to see in the future, and that is not a problem for the IndiTreat technology. We haven't been here quite as many years as some of the previous speakers, but anyway, we have been there 15. This is actually our 15-year anniversary this year. So we have built a lot of know-how and IP. We have a strong IP estate. and that relates to the full process of this IndiTreat technology. So that's both the 3D microtumors, a system with gel arrays, analysis, and what have you. And we convinced that makes the IndiTreat as pretty much the state of the art in functional testing. So that's the technology. Then I mentioned to you that we have some clinical data, and that's what I'm going to introduce here. We started a trial in 2017 in metastatic colorectal cancer. It is an investigator-initiated trial, and why I'm saying this is just for you to understand. This is a trial where we are not sponsor. It's the hospital that is the sponsor. And when being sponsor, you have complete control over protocol, endpoints, how it's conducted, data analysis and publication. And in many ways, it's a little bit maybe dangerous for a company to give that to a hospital. The upside is if it works, then you as a company are pretty much like a third party because it's all controlled by the hospital, the future customer. The ones that we have had to do this is University Hospital Vejle in Denmark that have been appointed the best cancer hospital in Denmark, I think 50 years in a row. It's only cancer at Vejle. And it is a prospective interventional trial. That means that the patients have been treated according to our test. And the endpoint, we had one endpoint, primary endpoint in this trial, and we wanted to improve the progression-free survival, and that means that how long time does the patient sort of have a stable disease until it starts to develop again, progress again. And standard of care after 8 weeks of treatment is that 20% of the patients will have stable disease. So unfortunately, it's not that high. The clinicians at Vejle said, okay, you need to show us you can do double that number. We want to see that such a new technology can actually add 100%. So 40% of these patients should, after 8 weeks of IndiTreat guided treatment, have stable disease. Did we succeed? We actually did. And this, maybe we should have had breaking news here because this is something that all the data actually came out here 3 or 4 days ago. The data have been submitted to ASCO, the American Society of Clinical Oncology, the biggest cancer meeting globally. And they published our abstract, or the accepted abstracts here a few days ago. So that's the second good news. So not only did we reach the 40, we actually moved it up to 50%. So Fernando, now it's your turn to sell it.
Yep. No pressure. Yeah, so the good thing is that what we are seeing is functional testing as a concept has been around for many, many years. And as Ole said, it has been used in microbiology, for example, very successfully. What we are seeing is that now it is becoming mature to go outside of the research field and actually into the clinical field. These are just examples, but you see here the publications, how number of publications that are going out with, for example, cancer organoids, which is a generic way to talk about these replicas in the tumors. Last year, the National Cancer Institute at the NIH in the US actually organized a webinar specifically on functional precision oncology for cancer treatment selection. So it cannot get more specific than this in terms of clinical use of functional testing. More recently, a couple of months ago, the American Association of Cancer Research in their annual meeting actually held a major symposium called Functional Precision Medicine in Cancer. So the thing is, this is really becoming now mature to transition from a research technology into the clinical field. And the good thing is 2cureX has been for 15 years, as Ole said, preparing for this moment, so to speak. So the company was founded in 2007, and since then it has been fine-tuning the IndiTreat technology, making it more robust, reproducible, building intellectual property protection around some of the key processes in this, and most importantly, well, of course, building a strong financial position by being listed in the Nasdaq North, and having an experienced management team. But then, most importantly now, recently, is building all this clinical evidence that brings us to the point where we are now. So we are just in the point where the clinical evidence is ready, and the whole concept of functional testing is getting now more familiar for practicing oncologists. So what's the next step? The next step is obviously market access. So some people might think that, oh, now we have the clinical evidence, that's the end of it. No, actually it's the beginning of it, because now with this clinical evidence that we have now we have to enter into all this reimbursement and clinical guidelines world. A lot of common themes with the previous presentation. So we have identified, actually we prepared a matrix with all the countries in Europe, and we rank them based on size of the market and complexity. The problem with reimbursement and clinical guidelines is that there is not one single European pathway. Each country has its own system. So you have to go country by country. So what we did is we looked at the complexity of each country in Europe and then the market size, and we selected the countries where we have the largest size and the lower complexity, so to speak, to make it easier. So we have done that, and we have prioritized now finding distributors in these markets. I will go to this in a moment. There are two markets which we know are very complex in terms of reimbursement, and one is UK and the other one is Germany. So in these two countries, what we have done is we have appointed consultants who are helping us now prepare all the documentation that is needed to file for reimbursement in those countries, and also in France. France is somewhere also in the high level of complexity. But this is a long process. This is a process that can take years, actually. There are other countries where we expect to see the uptake much faster. So we are working on this, and we are also working on the CE marking to comply with the new IVD regulation. And that was a question that Ole made before to another presenter, because this change from IVDD to IVDR is extremely important, and it's changing a lot of things. So the kind of documentation that you have to submit to the authorities to get CE marking under IVDD is one level of complexity. Under IVDR, it will be a different level of complexity, and all products in the market have to be updated. So we are preparing for that. And how are we going to grow in the future? So what are our vectors of growth, so to speak? The first one is related to the product portfolio. This product that we have now, this clinical study that Ole explained, is for one specific application, which is third-line metastatic colorectal cancer. In the lifetime of a patient of colorectal cancer, the oncologist will have to make treatment decisions at multiple points in time. And what we are doing is we are developing specific applications for each of these points. So now we have the third line, and this helps the doctor identify what are the treatments that are suitable for this patient in third line. But we are already working in developing products for first line, and eventually we can find products for neoadjuvant chemotherapy and other things. So within the scope of colorectal cancer, we can develop multiple tests to be used in multiple points in time by the oncologist. And then, of course, we can expand beyond colorectal cancer. So we have ongoing studies in ovarian, pancreatic, and other tumor entities. So that's one way how we are going to expand is through a continuous development of the product portfolio. The other one is obviously geography. So again, as I mentioned, we have now prioritized for 2021 Initially, in the original plan, it was Nordics. But then we did this research, and now with the strong data coming out of this clinical study, we decided to also accelerate activities in Poland, Italy, France, and then Spain and Portugal as one single country. So we are actively discussing with distributors in these countries to launch the product in all of these countries more or less at the same time with the evidence that we have now. Again, these are countries where these reimbursement schemes are more accessible than in other countries. Okay. And then the third roadmap is related to automation. So today, IndiTreat is offered as a service. So we have our labs here in Copenhagen, and then we receive the samples, we test the samples here, and we issue a report that is used by the oncologist to make the decisions. We have an ongoing project with Hans Schicker Institute in Freiburg to automate significant parts of our process. Okay? Now, when we are able to automate this, obviously we improve our efficiency, but also it makes it much easier to open labs, additional labs, so we can open a second lab, a third lab, a fourth lab. This enables our geographic expansion. So one of the questions that we get recurrently is what happens to the US market? And the answer is nowadays we cannot ship samples from the US market to our lab here to test them, because we are shipping live cells, so we have a certain time span that we have to keep. But if we want to open a lab in the US, then we want to make sure that the process is exactly the same as we are doing here, etc. And this reproducibility is much easier when we have an automated system. So this automation project, which will enable this geographic expansion for us. So these are the three growth vectors that we are working on: geographic, product portfolio, and the automation of the process. A couple of words on the financial highlights. We have just released our annual report, I think last week. And basically, we have nowadays in cash. Last year, we had a directed issue. So we have now in cash something like 5 times what we used in 2020 in operational cash usage. So obviously, I'm not saying that we have money for the next 5 years, because we are going to accelerate our consumption of money as well through these activities in the different markets. But we have a quite solid financial position. This has complemented with, so this directed issue had associated a warrant program that has been finalized last week, I think. And we have raised 31 million Swedish krona additional. And then more details are available in the annual report. So in summary, and then we can move to the Q&A session, we are convinced that functional testing is the next wave of precision oncology. So biomarker testing has led in the first 25 years of precision oncology. We think now the next wave is going to come from functional testing. 2cureX is leading the space of functional testing. We have a solid and protected technology, we have a product in the market, and we have the strong clinical evidence to support that. We have a clear go-to-market plan. We are working on country-specific strategies, reimbursement, getting the product in the guidelines, getting the product to be used by oncologists in routine. And we are getting for the regulatory change in 2022. We have a good vision of how we are going to grow going forward. And we think at this point, we offer an attractive risk profile in a business that has a huge potential for growth, because the size of this market is going to explode. So with that, I think we are ready for the Q&A.
Even though you are 2 speakers, you will have the same time question. Yeah.
Yes. So a technical question, since you are handling cancer biopsies from patients, is there a challenge to keep these cells alive properly in the in vitro environment, and is that something that is a continuous struggle to try to get streamlined efficiency and avoid variability between sample to sample?
that certainly is an issue.
Ole, will you repeat it?
Oh, sorry, yeah. So the question was that you are using live cells and keeping them alive during the sort of active screening process. Is that a problem? And the quick answer is yes. I mean, these are not only are they live cells, they are primary cells, and that means that you really need to nurse them, and that's we've used 15 years to do that, to create a system that these 3D, and that's very, very important if you know about cell biology. Normally you use flat biology, so you have them sitting on a surface, but that's not how solid cancers work. I mean, they need to attach to each other and then need a certain environment, and we have that in the system. So it is an issue, but it is handled here. As soon as we have these tumoroids formed, then they will almost in all cases start growing.
Very cool.
Start with the lady.
Okay, how many patients were included in your phase 3 trial?
Yep.
How many patients?
Okay. In this trial, we were allowed to include up to 90 patients, and 45 of them were actually included in the trial where the treatment was guided by IndiTreat.
Lars?
I was thinking about this ex vivo test. When you find out that that a drug seems to be effective, what's the predictive power of that? I mean, when you have to treat the patient in vivo, you could say, right? That was one question. And another question is, what I'm thinking about is that isn't this technology more better for small cancer indications than bigger cancer indications? Because if you take the bigger cancer indications, you get a lot of data by the time you get a lot of data about what kind of drugs are most suitable for different patient types.
So, if I take, okay, so the last one was, is this technology not better suited for a smaller cancer disease than a big one like colorectal cancer?
Yeah, because the information, you get a lot of information on the big cancer types, right? Because there are a lot of patients.
Absolutely, and I would say no. If you look at colorectal cancer being the second or third, depending on whether it's women or men, cancer, there we have standard treatments that are based, as you say, on an overwhelming amount of data. Still, we see that in first line, more than half of the patients do not respond, and we need to change treatment. So even there, it's a big problem. So I don't see that it's better suited for smaller cancer entities. Where you're right in that if there's not that much information, then you can say, then this, I mean, there's no biomarker or anything, then this is very suited. And I think maybe if I can sort of change it a little bit, we are seeing a lot of approved drugs getting into the market. FDA has, over the last couple of years, approved more drugs than they've done for a very long time. And there's a lot of criticism to that, in that the clinical evidence is becoming smaller and smaller. So they are approving drugs because there were a lot of criticism that not enough drugs came in. Now a lot of drugs come in, and we do not have that much clinical evidence. And there, such a test will be of value as well.
Morten?
Just to follow up on the question here about the live cells. You said you had 90 patients coming into the trial, but something like 50% were only the live data. What happened to the 50%? What did you get any response on the microgenes on that?
Morten always comes with the nasty questions. So, but that's a very fair question in that you can say we had 54% of the biopsies that we received we were able to make an entry test, and why wasn't it not 100? And the quick answer is that we were taking liver biopsies of heavily pre-treated patients. They had got all the standard treatments available, but were still progressing. So, for us to be able to make this test, we need viable cells. So actually, only about half of the biopsies we received had living cancer cells. So if the living cancer cells are there, then the success rate is 80-90%. And maybe I should say that for these late-stage patients, a lot is happening with regard to improving the biopsy procedures. We are seeing new technologies coming, and we are following that, and that will increase the number.
The fact that 50% of the patients, samples from patients can't turn into a viable product, will that be a hindrance for taking the product commercially?
Oh, now, now it's for me. Nice. No, as Ole said, first, the third-line product is the most challenging one in terms of the quality of the biopsy that we receive, because these are the most heavily treated patients. When we move into second line and first line, it's going to get easier, first thing. Second thing, it's improving because the techniques available for biopsies are also improving. But we are actually in discussions with a couple of healthcare authorities in a couple of different countries, where the approach we are taking to that is related to resharing. And I think probably you're all familiar with the concept of resharing in healthcare. So we are discussing whether there is a way where we would get our payment for the treatments based on patients where actually the thing is successful. Obviously, the pricing will have to include that risk, but it's a risk that is easy to calculate. So we are having this kind of discussions also, exactly to avoid that this topic about the viability of the samples that we receive might slow down or prevent the uptake of the test.
Continue here. You were first.
Yes. Two parts of my question. How many customers do you have now who sends a test to you? And what is the price for a test, and how much of that price is earning?
Okay.
3 questions.
Yeah. So nowadays, all the samples that we have received over the years are related to clinical studies. Okay. So we don't have hospitals who are sending samples outside of clinical studies. Okay, this is the first thing. The price of the test, let's say the end price is going to be in the range of 3,000 euro per test. Okay, I don't think, and that will include then the margin for the distributor and our gross profit. But I think I would rather not disclose that at this point, especially because we are in a competitive environment, and I don't want to give too many clues to our competitors.
Back to the 50%, could it be that there are simply no drugs working in third-line treatment, that the patients will not respond to anything?
No, because that 50% or 54%, where we were able to do the test, say that was the remaining there, the cells were dead. So that was the reason why there were no screening. But actually, with the 54%, not only were we able to make the tumoroids, the test, we were also able to provide at least one treatment to the physician. So
there's always one treatment?
Always one treatment, at least of these, and you have to compare to the genomics where it's 11%. I mean that's what, and genomics will not work in 50, where it definitely not respond in 50%. That's 11%, and so yes, this is a very difficult patient population to work with. but we just took the worst and most difficult to begin with, so it will only be better. I also have to say, we have had absolutely no problem recruiting patients. All patients have said, yes, I'm prepared to do it, because there's not that much with it. So you can live with, say, okay, it didn't work, we may have to take another biopsy. They will do it.
There will be 2 questions back. One for you, Jørgen.
Yes. So you have a question a bit on the science side, but since you do get all these samples from patients and you do test them for various drug combinations and see what works against them, that is a really potent source of future information that you could perhaps use for developing something similar that would be more potent in the future if you combine genetic screening and such. Is that on the agenda for collaboration-wise?
So the question was that we are generating a lot of information on sort of how these different drugs work in different patients, and is that all that information?
You can create a fantastic library of patient material in this manner. Do you save material from every patient that you get samples from?
I will just ask a small bit, and then I will ask Fernando to take the other, because I mean, today we are using that information to provide a reference data set to the clinician. So the clinician will not only get sort of how is your patient doing, he or she will also get a panel of how similar patients respond, and they are really happy to have that. So that's how we use it now, but using such a database in the future.
Yeah. Actually, let's say the vision is a combination of all this data that we are generating on which drugs work or don't work in different patients, but very importantly, this in combination with a biobank of those samples. So this is, as you say, this would be a unique resource for research, because when you know which patients responded or not responded to a certain drug, then you can go backwards. I was talking about the biology before. Then you can go backwards and try to find what are the molecular profiles that would explain this difference. So absolutely yes. Now, databases and biobanks are highly regulated and increasingly regulated in Europe, and therefore, before we make any claims on potential commercial activities around this, we have to build that according to all the rules and regulations. But the answer is yes. This could be eventually the most valuable asset of the company at one point. Yes. And by the way, with the artificial intelligence envelope, if you want to do this analysis, and this is where, going back to the first slide when I mentioned that Kenneth is coming from an AI company, that's also where that experience can help us a lot. Not only the experience, the network and everything that he has there. So.
One final and very short question is from Morten.
Maybe a bit of a softball there, but if you guys have been a standard biotech drug development company that had a product that could increase PFS from 20 to 50% in colorectal cancer last line, you would have been late breaking on ASCO and all of the news. Why are you traveling around with a few $100 million market cap when you have such a great opportunity? Is it something you're missing, or is it the market missing something? You guys
Okay, I think first, I think.
picked the question.
Yes, I think the question is if we would be a biotech with a drug in early stages that would prove a change from progression-free survival from 20% to 50%, we would be kind of swimming in gold, more or less, right? Or in dollars, probably. And why is that not the case? That's the question, I think. And here, I mean, it's just opinion that I can provide here. But I think investors are much more familiar with the steps in a biotech company. So, and going back to the first presentation that we had today, there are very clear milestones and drivers of growth of value of the company, et cetera, et cetera. So everybody's familiar with when the phase 1 study has finalized and it's positive, then this is a check. Then phase 2, then phase 3. In diagnostics, this is not so clear, I would say. So it's not that obvious for investors that this has been a milestone that actually has a tremendous impact in the risk profile and in the potential value of the company. I think that's not that as evident as it would be in a biotech company. The second thing also is, as Ole said, unfortunately, we don't own the communication here totally because this was an investigator-initiated trial. So probably if we would own the data and we would own the timing when we release the data and how we explain the data, etc., it would have been done differently. Here, we were forced, because of market regulations, we were forced to disclose that the endpoint had been met, but we couldn't go into any details because of the ASCO restrictions, so we were kind of trapped in the middle of the ASCO restrictions and the market regulation obligations. So from this point of view, maybe our communication has not been optimal, but we did what we could given the circumstances. I would say these two things probably, but again, it's just an opinion.
Fernando and Ole, thank you very much for your presentation.
Thank you.